All right. Good morning, good afternoon, good evening, everyone, wherever you are, to this new edition of the Worldwide Neurodevelopment Forum. Today's guest is Tomomi Shimogori from the Riken BSI in Tokyo. She's actually, I was realizing, you are our easternmost guest to date. We'll have a guest from Australia in a few weeks, but you are the easternmost one, and I'll start by thanking you for that because it means it's 11 p.m. right now.
12 a.m.
Oh my God, it's even worse than I thought. Okay, so 12 p.m. And not only is Tomomi going to present late, but in fact, she pre-recorded her presentation because for some reason, the connectivity between Tokyo and Geneva, at least, is somewhat unpredictable right now. So I'll be sharing my screen, and she pre-recorded that. At least you don't have to present it twice, Tomomi. Anyway, so I'm really happy that you accepted this invitation, and thanks for accommodating your schedule to the rest of us. A few words of introduction for those of you who are not familiar with Tomomi and her work. Tomomi did her postdoc in Elizabeth's Grove lab in Chicago. She's been independent researcher at the Riken BSI since 2004, but before that she was in Chicago, where basically she did a beautiful piece of work there. In my view, really one of the most influential papers in developmental neuroscience to date, where she showed that overexpression of FGF8 ectopically could reassign the regional identity of a cortical region and actually reroute thalamic axons towards a pseudo somatosensory area. Since then, she's been building on this work, but also expanding in other areas. I would say I see your work, Tomomi, as really at the interface between thalamus and cortex, with some very insightful views also on how cell intrinsic transcription factors such as BTBD3 play a role in dendritic complexity, but also focusing on the thalamus itself and figuring out how this complex structure comes about in terms of nuclei and connectivity. More recently, Tomomi has been focusing or shifting or expanding her attention towards other animal models than the mouse, and in particular the marmoset. And I think this is going to be the main focus of her talk today. So Tomomi, really, it's a pleasure to have you. Always nice to see you. Welcome. So I'm not going to say the stage is yours. I'm going to say I'm going to share my screen, which is a bit less grandiose, but still comes down to us having the ability of hearing you safely. What will happen is that once Tomomi's talk is over, we'll switch back to this mode, and hopefully the connectivity will be good enough for Tomomi to take questions and answers. So I think here, yeah, and of course it's not showing what I was wanting it to show. Give me a second. Why is this, guys? We trained this so many times, and now I'm not seeing it. All right, it's here. So I'm sharing my screen. Let me refresh. I'll be back in 10 seconds.
I'd like to start thanking Jenny inviting me here to provide me the opportunity to introduce our recent project. So to understand the human brain is our ultimate goal. So especially human brain function and dysfunction, and also how it develops human specific brain region and function is something what everyone's probably interested in. To understand human brain, of course, we want to use human brain itself. However, there's a lot of technical limitation to use human brains. So we have to use different species that we can manipulate or look more into details to understand human brains. So to do that, to do the comparative analysis is actually very important things to do to find out what is similar and what is different among each others. This is the figure which I took from Leah Kravitzer's review, showing that a lot of conserved brain region exist in mammalian brain, cortical brain regions. For instance, the region color in blue is primary visual area, yellow is primary auditory area, and also red is a primary somatosensory area that you can find in all of these different species. However, the size, position of these individual regions are slightly different among each species. And then using these conserved brain region, we can probably understand what kind of similarity they have or what kind of differences they have to understand finally the human brain function. However, even we say that there are conserved brain regions such as somatosensory region, if you look more into the detail, you actually can find that within those conserved brain region, the connectivity and also anatomy are quite different. Here's an example of 4 different animals on the somatosensory region, showing that how different they are. So it depends on what kind of peripheral they are using to collect more sensory input. You can find that the size of the somatosensory region and also the connectivity of the somatosensory region are quite different. So even in a conserved brain region, if you're looking at the connectivity and their morphology, you can actually have difficulties to understand how these regions are developed or evolve in different species. So to understand how similar these brain regions are or how functioning these conserved brain region, we should have another way to understand or dissect these differences or similarities, which we think the molecular identities can also help to understand these questions. So this is an example of gene expression pattern in common marmoset cortex and mouse cortex. So these genes represented here are example of layer-specific marker genes. And if you compare the neonate common marmoset and equivalent stage, which is about P20 mouse brain, you can find similar distribution of these layer marker genes. So we can use the molecular markers to understand how these equivalent regions look similar or different. So let me introduce this database which we've been working on from 2016. So this is the database of common marmoset gene expression, and this project is run by BrainMinds. This is the Japanese nation project, which is trying to develop common marmoset database, starting from gene expression, connectivity, MRI data, and also like behavior analysis. So you can visit to the BrainMinds website to find out what kind of activity we are actually doing in more detail. So as one of BrainMinds member, as I mentioned, we started to work on gene expression analysis on the common marmoset brain, and we have established this database, which is open to public from 2016. Currently, we have over than 2,000 in situ registered on this website, and we are actively updating the data almost every 2 weeks. So this database is already open to public, and if you're interested in, I really encourage you to visit this website frequently. Okay, so then why we focused on common marmoset? So common marmoset is New World monkey, and to do these kind of molecular work, they have a lot of advantages. So first, their gestation period is really rather short compared to other non-human primates, which is 148 days. And their high productivity, which means like they actually give birth each time they're 2 to 3 offsprings. And their body size is relatively small, which means that it's very easy for us to handle them. They also develop rather faster compared to other non-human primates. They become sexually mature in 12 months. However, their life expectancy is like 10 to 15 years, which is probably long compared to their small body size. They also have very interesting behavior characteristic. For instance, they're monogamous, and once they decided to stay couple, they maintain their partnership till for the rest of your life almost. And what is also interesting is child rearing by both parents. So they basically, both parents carry their offsprings on their back, and they help each other to nurse all of those offsprings. Also, what is interesting is their older brother and sisters help rearing for their younger brother and sisters. So they're a very strong family unit. Here's how they look like when you have them in cages. So basically, you can keep multiple marmoset in one big cages, and they actually interact to each other heavily. And you can find that they can actually use their hands to search food, for instance. There's a little treat inside of this paper cup. So now they're trying to open it and find the treat. They also share food. This case, this is not sharing. This one is the food is taken by their friends. But basically, they have very interesting behavior, which is a little bit different from other non-human primate model animal that we are using in the lab. Okay, so this is another information to give you how their neurobehavior developmental time course look like. So here, once they're born, this is like a week of ages, and they have multiple different time point of their critical period. For instance, their visual critical period is sometime between 1 to 2 months. So it's rather faster compared to different species because their eyes are already open at the time point of the birth, and they are already responding to the visual cue. And then actually the locomotion critical period comes after that. And the weaning is between 3 to 6 months. It really depends on their individuals. And they become sexually mature, and then they start to interact to each other, and then slowly they start to mature till up to 2-year-old. Here's just a comparison of how human brain development, the time course of human brain development, and you can find that they have sort of similarity of the time curve, although of course the human actually have a little bit slower, so this is the time course is month here, and instead of that, the time course of week here, but you can find some similarity among these two species. Okay, so basically, what we wanted to do was using this database is we wanted to find out the molecular profiling, which shows similar to common marmoset and mouse, which is the most frequently used animal model in our laboratory. And also, we wanted to find the major differences of gene expression to find a different cell diversity in a specific brain region. Since we have more than 2,000 in situ data in our database, we thought that we have to have some tool we can find gene expression systematically. So we developed this tool called GEPS, Gene Expression Profiling System. So basically what this system can do is once you point the specific brain region, it can find the gene which is expressed in this specific region. So here's what's happening behind the system is we put the grid on every single in situ data, and within the grid, the system picks up whether it has in situ signal within that grid or not. So if there are positive signal, this is how it looks like. You cannot see this on the website. However, once they have this signal or this positive signal within the grid, then it shows as a gene expression. So you can get the list of the gene. This is actually something similar to the Allen Brain Atlas called AGEA. So here's how it looks like in their website. Once you point a specific region of the mouse brain, then it actually picks up the genes which are expressed within those regions. So using Marmoset GEPS system and Allen Brain Atlas AGEA system, we can find gene expression in a specific brain region and can compare those lists. So to give you some example, this is the result, the search result that we have done in common marmoset primary visual cortex. So here you can find there's a specific brain, gene expression in the primary visual cortex. And once after you get the list of the genes, you can compare with the mouse brain. And once again, this is the same set of the gene shown in above. Then you can find they have very different gene expression in mouse brain like these. So we can do this kind of comparative analysis. And here's a result of the gene ontology analysis, which you've done in the gene expression specific in the primary somatosensory and the primary visual cortex. If you look at here, you can find what kind of molecules are expressed in the primary somatosensory in a conserved manner or common marmoset specific manner. I want you to focus more on the primary visual cortex. What you can find here is it is quite very different compared to the somatosensory cortex, especially in the primary visual cortex. They have less catalytic activity gene expressed as a conserved gene, so which tells you that the primary visual cortex have a very different gene expression compared to marmoset and mouse, even that region which is set conserved brain region. So here then, if the primary visual cortex is so different, what about the presynaptic neuronal population, which is mainly from the lateral geniculate nuclei population in the thalamus? So we have done the similar kind of analysis using common marmoset GEPS system and Allen Brain Atlas AGEA system. So this is the section that has or contain marmoset lateral geniculate nucleus next to that, which has the ventral basal thalamic nucleus. And when we have done we have analyzed the gene expression specific to LGN or specific to BB. And these letters, which are in magenta, mean that the gene expression is specific to common marmoset. When the letter are in blue, which is not shown in this slide, which shows in the next slide, the blue labeling means that it is mouse-specific expression. When it is black, expressions are conserved in marmoset and also the mouse. Okay, so here, like these two, which has specific expression in marmoset ventral basal thalamus, you can find these expressions specific in the ventral basal thalamus, which means that these genes are not expressed in mouse ventral basal thalamus. And here, these two genes are specifically expressed in marmoset LGN. However, these genes are not expressed in mouse LGN. Okay, so next, there's an opposite case. So for instance, here, if you look at this gene, the expressions are specific, it's not specific, sorry. The expression you can find in the ventral basal thalamus here, but if you look at the LGN, this is, which is in blue, means this expression is specific in mouse, but not evident in marmoset. And also here, this gene, which is FA5, there's a specific expression in mouse. However, it's very difficult to find its expression in marmoset. So these cases is basically whether mouse expressions are acquired specific in mouse, or it is lost specific in the marmoset. So there's both case, if the gene expressions are not conserved, there are two cases: either one species obtain expression specifically in the specific brain region, or the gene expression is specifically suppressed in that specific region. So there are both cases. Of course, we cannot tell which is which at this moment, but at least this GPS-AGEA comparative system can reveal these region-specific gene expression, or the gene which is not expressed in those specific region. Okay, and here I just want to show some example, like in situ actually is a really powerful technology to show spatial gene expression. So here's a case of the gene which we found specifically expressed in MGB, medial geniculate body. These two here are expressed in MGB, as you can see here, specific in marmoset, but the expression is not just simply specific in marmoset MGB. You can find that there's a spatial specificity. For instance, this NTN1 gene, which has lateral strong, and then the medial part actually there's no expression in the medial part. ADRI2A, which has more dorsal expression, but it doesn't have expression in the ventral, or you can say like there's a gradient expression, dorsal strong to ventral weak. So this is also the gene TNNT1, which has conserved expression in mouse and marmoset. However, when you look at the expression within the MGB, while mouse has ubiquitous expression in MGB, in marmoset they have ventral strong and dorsal on the dorsal weak expression. So you can get these kind of like a gradient expression or spatial expression within the specific nuclei by in situ hybridization data. Okay, so here once again, this is a gene ontology analysis within these three different thalamic nuclei. I'm showing that how different or how similar they are by the specific molecular function. Once again, I want to focus more in this visual circuit in LGN. What you can find is they are very different, especially in the common marmoset specific gene profiling compared to other BV and MGB, which might mean that the visual circuit actually have very big differences compared to the mouse visual circuit. Okay, then why so visual circuit are so different, even they have conserved brain regions such as LGN or primary visual cortex? You probably have already noticed that, of course, the connectivity have very large differences among these two species. For rodents, like those animals which have their eyeballs on the side of their faces, there's a less ipsilateral projections happens, and also in LGN, they actually their structure are quite rather we can say that it's rather simple. On the other hand, like primates, those eyeballs are placed in the front of their faces. They have lots of ipsilateral projections. Therefore, also in the lateral geniculate nucleus, these input from contra and lateral are nicely organized in the layer formation, and these input are never intermingled, and then they reach the primary visual cortex into the ocular dominance column. So here, this is the comparison of the morphology of rhesus, LGN, and marmoset. As I mentioned, in LGN, they have beautiful lamina formation, and also they, compared to the marmoset, even you can find it's a little bit more simple layer formation. However, you can still find this beautiful lamina formation even in marmoset LGN. So these lamina formation in the LGN, you can find not only these lamina, find the magnocellular portion, also cellular portion, which is more dorsal side of the LGN. And these magnocellular and primocellular are known to have different connectivity in the primary visual cortex. So here you can find the magnocellular, which is projecting to the layer 4 CA alpha of the primary visual cortex. Compared to that, parvocellular portion are projecting to layer 4C beta of the primary visual cortex. So here the different connectivity actually exist in the LGN, which is not seen in the rodent LGN to primary visual cortex. So we thought that whether our GPS can find these molecules specifically expressed in magnocellular portion or parvocellular portion. So and here's the result. You can actually have that kind of spatial resolution using GPS and can find these magnocellular specific gene expression. Here's just an example. And also you can find the parvocellular specific portion like this. And just as one of the example, this NPY2R, which is the parvocellular specific expression in the marmoset, we have also done the comparison with the mouse equivalent stage, which is about P14 to P28. You don't see any expression of NPY2R in mouse LGN like these two. However, this is not the in situ error because you can find conserved expression in their hippocampus. So this means that this is a very specific gene expression differences in LGN among these two species. So then I was just showing more like a nucleus-specific gene expression. We wonder whether GPS can also find genes which have like a gradient expression in specific, non-human primate specific regions such as pulvinar. So these days, pulvinar equivalent and also exist in mouse, which is LP. However, their components are quite different compared to mouse pulvinar and pulvinar. That you can find that we can divide pulvinar to lateral and and then I'm going to go into detail, but these are just showing that using GPS, you can also find these genes
Hello, is this working or not? I see we're live. Should I continue to share the screen? We lost Tomomi live. Ah, voilà. Thanks, Zoltan. So this is unusual. There's always something new because for some reason, my camera looks like this now. What I'll do is, yeah, exactly. Only see sound, yes. So I'll continue to share the screen. I'll rewind 2-3 minutes, and we'll continue. And I'll try to. Well, I'm hoping Tomomi will join back. So I'm going to share my screen. Voilà. So this should work. Maybe Simona, if it doesn't work, send me a WhatsApp or something.
In the subplate and also in the mouth, like a 3-month-old. And we actually think that they actually pulvinar equivalent region also exists in mouse, which is LP. However, the size and their components are quite different compared to mouse pulvinar and non-human primate pulvinar. That you can find that we can divide pulvinar into the medial to lateral and lateral, as you can see here. And then I'm not going to go into detail, but these are just showing that using GPS, you can also find these genes expressed in the specific region of pulvinar. Okay, then as I mentioned at the beginning, our ultimate goal is to understand human brain. And here we were looking or comparing gene expression with the common marmoset and mouse. Our question is whether we can also use marmoset and do the similar comparative analysis using human tissue. So currently, the human gene expression database we can use is the Allen Brain Atlas human brain database. And the youngest age in the postnatal stage we could find is like a 3-month-old. And here to do the direct comparative analysis, we use their primary visual cortex. And this is a little bit busy slide, but I'm not going to take you all through it, just going to give you some example. This is the only one gene that we actually found. The common marmoset, only the common marmoset show the very different gene expression called CCN2, CTGF. That's how it used to be called, known as a subplate specific marker. So here you can find in the human brain, there are strong expression in the subplate and also in the mouse brain. However, the gene expression or the CCN expression in the common marmoset is evident in layer 5, and there's a really strong expression in upper cortical layer. Okay. However, beside those, that gene expression, what you can find are, like, for example, these GAP43, MBP, NNAT, and PDYN, which has similar expression with human and common marmoset, and mouse have very different expression pattern. For instance, near netin here, you can find there's a expression of layer 5, and there's scattered expression in upper layer. However, in the mouse, you can find expression everywhere in the cortex. PDYN, there's a strong expression layer 5 again. There's a little bit of expression in upper layer in common marmoset. However, in rodent, you can find, again, strong expression in upper layer everywhere. So here, this is a very small number of the examples, but you can find lots of similarity with human and common marmoset gene expression, and the rodent seems to have a little bit different compared to these two different species. Okay, so after doing lots of comparative analysis with rodent and common marmoset, we have also realized that if we can do a little bit larger scale comparative analysis, we thought that it's going to be also very helpful. which the larger comparative analysis, I mean, is like those gene expression within those big regions, for instance, like a cortical areas. And when we want to find the marker gene, which is specifically expressed in the specific cortical areas, of course, our data are in coronal section, and you can just have to find these genes which have a specific expression with the specific borders like these, right? But when we are talking about the cortical area, of course, those areas are not in 2D, right? You have to look into the 3D. So we thought that if we can make these 2D images back into 3D, we can get better spatial resolution to understand how these genes are expressed. Also, we thought that if we can overlay multiple gene expression with probably we can find more different brain regions in case like if they have overlap or no overlap. So to do that, what we had to do first is we had to collect all of these in situ data, and of course the strengths of the gene expressions are quite different. So we made it into black and white to adjust the strengths to every individual sections, and then we put it back into 3D. So basically we stack all of these 2D images all together and then put it back into 3D. So then also what we have done here is because we wanted to look at the gene expression in the cortex, and also wanted to compare multiple numbers of gene expression. However, these genes are not always expressed in same layers, right? So it can be distributed to different regions, and in case if they're expressed in the different layers, it's a little bit difficult to overlay those gene expression. So what we have done here is no matter which layer they are expressed, we projected all of these expression into one layer. So here is the example image that if these are the brain cortical plate, and no matter where they are have expression here, we just projected the gene expression in one layer. In this case, this is on the superficial layer. So all the gene expression in the different layers are now in one layer. Okay, so then we can put into the MRI image, so you can get the image, the gene expression in 3D. So here's the example of this, how this cortical area projection mapping look like. So this blue gene this is the dorsal view of this blue gene expression pattern. You can find their expression in dorsal and lateral like this, and you can actually overlay another gene expression. So now this is in the second gene expression in red. In this case, if the red and blue, so if it shows purple, this means that they have overlap expression. So here we also can put the third gene on top of it. Now this is in green. If they have like a gene expression overlay with 3 of these, now it started showing like yellow region like this, which just started to show like this, which we couldn't find in these single gene expression. And here in the lateral view, this is more interesting. You can actually start to see the primary visual border, secondary, and also V3 and V4 like this. And there's V5 somewhere around here. So using this cortical area projection mapping, probably we can find more cortical brain region that cannot be found by the single in situ hybridization, especially in 2D section result. Okay, so here this is my final slide. And so I just showed you we can do quite a lots of different comparative analysis using our database and also GPS function, and also with the cortical projection mapping you can find lots of new brain region that cannot be seen by the single in situ hybridization. We also thought that the reason why we are using this non-human primate is because we also want to understand the human brain dysfunction. So we decided to look at those gene expression, especially related to developmental brain disorders, and also wanted to compare with the rodent, because sometimes the making or generating the knockout animal or transgenic animal for these disease risk genes doesn't show any phenotype that is similar to human disease cases. So we thought that it might be because the gene expressions are quite different with rodents and these like a human cases that might be causing these different phenotypes. So here, again, this is a little bit busy slide. I'm not going to take you through the whole thing, but this is the one thing I want you to focus. This is just an example, CHRNA2 expression that we have looked into the cortex, which is in pink here, and in green, it's in the thalamus, and this yellow is in the striatum. In the cortex, we look at the somatosensory, visual, and auditory. And this means that there's no expression in mouse and common marmoset. And also no expression in striatum and marmoset and mouse. However, when you look at the thalamus, you can find that this gene is expressed only in marmoset ventral basal thalamus and marmoset LGN. So here, in case when you generate the knockout of CHRNA2 in mouse, since there's no expression in these three different brain regions from the beginning, you may not see any functional defect, which probably you can see in the human cases. So again, here behind this thing, you can find lots of genes like this. We did a lot of analysis. These columns or rows that you can find in the darker color is either expression specific in mouse or specific in marmoset. So basically, you can find that there are a lot of gene expression which is specific in rodents or specific in common marmoset. So once again, so if you are looking for the gene function, especially when you want to look at the behavior using these transgenic animal, you want to be careful to test how these gene expression look like in different species. Okay, so I hope I could convince you that how important it is to do the gene expression comparative analysis with different species, because even in the conserved region, there are quite diversity of the gene expression in different species, which might be causing their different function and connectivity, and also which cause a different behavior. So I would like to thank all of my team members, which is actually pretty much all of them are involved in since this is a big project. And also I'd like to thank one of my collaborators, Henrik Skippe, who is also in our institute working on the image processing and also helped a lot about the vertical projection mapping. So thank you very much for your attention. I'm happy to take questions.
All right. This is good. Let me stop the screen sharing. Voila, I see you're back, Tomomi. So can you hear me?
Yes, I can hear you.
Oh, it's actually working. I'm going to stay since we seem to be having some bandwidth issues, I'm going to stay small there. I'm a bit frustrated because it's my weekly opportunity to wear a shirt, but you've seen me. Thanks a lot, Tomomi, for this talk. Also, thanks a lot for your foresight. I mean, it was very very elegant and delicate of you to think that there might have been a problem, and so. But I think we managed it, and everybody got access to your talk. So thanks again. So. While people are I mean, you're seeing the side of the screen there, right? It's this awkward thing where there's these claps. So people in the audience, please do feel free to ask your questions in that little tab on the bottom. In the meantime, while people are typing, one thought that crossed my mind is relating to conservation of gene sequence, so at the DNA level, and conservation of spatial expression of the genes. Do you have any insights into this? Are the genes that are most conserved in terms of sequence at the level of the DNA? most conserved between the two species are also tend to have more conserved spatial expressions?
At the genomic level, I think.
Uh-huh.
Yeah, that is definitely possible. And of course, with our institute, we cannot discuss that level. And well, I didn't mention about in my talk, but the institute is not the only way to find the differences and similarities, like what you're also talking about, and like single nuclei, RNA-seq, and other things are also something that you probably consider about. So I think we have to do all of these different level of analysis to find when the difference is happening and where, kind of thing. So I'm not answering directly to your question, but definitely we have to test all of those different levels to really find out what is causing these differences and similarities, right?
But in terms of function, for example, can you tell whether transcription factors tend to be more conserved than, I don't know, receptors, or whether...
And molecular functions. I think it really depends on which part of the brain you're looking at. So like in the visual cortex, we found a lot of catalytic activity. Molecules are different, but if you look at different places, it totally shows different sets of molecules, which shows the differences. So we have to really be careful and look at the different part of brain in a different level.
Any insights into relationship between conservation, transspecies conservation, and developmental stability of the gene expression? You know very well that some genes tend to have dynamic patterns of expression during development. What's your feeling about how that reflects
Yeah, I totally agree with you. Yeah. So currently we were looking in neonate. So the data we have on our database are mostly neonate. But if you visit there, you can see that we have a plan to look at the different time point, like 1 month, 3 month, and 6 months old, and also like adult stage. I think you understand why, because of those time point that I showed you and for the developmental time point thing. Yeah, so that's also what we are thinking. And also on our database, we actually have done the next generation sequencing in a different brain region at a different time point, which is showing a dynamic gene expression profiling. So based on that, which has very different gene expression, we're definitely going to try in situ for those and see how differently they change their expression. But I think it has a lot of meaning, what they're doing at the specific time point. But again, we need to reveal the expression first.
Yes. So actually, I see that this was one question that Seth Blackshaw was addressing to you.
Oh, really?
Good point. Yeah.
Thank you, Seth. So how I'm sorry, how do I see those questions?
Well, you can click. I mean, I'm going to read them out to you.
Oh, there you go. Oh, thank you.
You can click on ask a question and you see them.
Yeah, found it. Thank you.
So we have Tomomi. We have Shuba here on the top. Hi, Tomomi. This is marvelous. Is there any special markers in the insula? Any hint? It's a special structure.
Well, I guess I should instruct you how to use the system, because so again, this is like a whole brains in situ database. So wherever the structure is, you can find.
What's the website, Tomomi?
So if you just Google MGA or Marmoset Gene Atlas, then you will hit it. So
okay, can somebody please do that and paste it in the discussion while we're
going? I see. That's
yeah. Yeah. Thank you.
And once again, this GPS thing, you don't have to type the structure in it. You just point where exactly you want to look at, and just click find the gene. Then it will give you the list.
Okay. Cool.
Okay.
A question from Zoltan. Fantastic talk, great data. Based on these comparative observations, what particular area of neuroscience should we consider using Marmoset as a model system? Yeah.
Well, I think everywhere, wherever you are interested in. Right. Well, I just this time, just to show the example how we can use this database. I was focusing more on the so-called conserved region, but I also look into like a prefrontal cortex. It's not like a straightforward that you can do to compare with the mouse, because they have very different part. But if you look a little bit more detail, in a marmoset prefrontal, you can actually divide in lots of small subdivision, which is very difficult to do in mouse brain. So basically, you can do a lot of analysis endlessly using this an analysis. And what part of the brain looks interesting? I think everything looked interesting to me.
All right. A question from Simona Lodato. Great talk, thank you. Would you have any insights on the genes that have divergent expression between human and mouse cortex? What is the frequency and the molecular features of these genes? Also, do you have any idea how non-coding RNAs behave instead? Thanks. It relates a little bit to what we were discussing before, but maybe the idea of the non-coding RNAs?
Well, then we have to go to the different level of the search on this, right? Non-coding, and well, it also adds like epigenetic search on it. Some of the part is taken care by this Brain Minds project, because we were trying to really go into more detail. At least for the single cell RNA-seq part, I'm gonna just started a collaboration with Nen Assistant to work on this. So we'll go into it. Non-coding RNA part, it's a really good part. I think we should propose this to start trying to do it. But currently we haven't started, but it's a really good suggestion. Thank you.
Question from Shingo Nakazawa. Thank you for the wonderful talk. What is your outlook on gene manipulation techniques for marmosets? Can we manipulate genes as easily as in mice, like in utero electroporation?
Okay, so I have to tell you, it's not easy at all. So we try to do in utero manipulation and marmoset, but first thing is basically the position of the uterus are very different compared to mice. So the mice that they are in sac individually, so they are separated to each other. But for the marmoset, they are most of the time they're twins, but they are close to each other. So it's really hard to see them since they're in the big uterus, and then it's not transparent, so you have to use ultrasound to first look inside, and also to position them. I have to say it's really very difficult to do. So probably the in utero electroporation is a little bit difficult. It might be possible to do in utero virus injection, but targeting might be the issue. And also economically, it's also a little bit difficult thing to do. So I won't say it's impossible, but it might be a little bit difficult to do. Yeah, but it'll be really great if we can do those kind of manipulation.
Hearing you say that, and having been in your lab and seeing you electroporate mice, I'd say if you say it's quite difficult, I would say it's nearly impossible. But
well, I don't want to raise the bar like that. To leave some possibility and hope for young, but yeah, it was not that straightforward. I have to be honest. Yeah.
So translate into super hard. Okay, one last question, maybe. Susan Faulkner, thanks for a great talk. In addition to the species comparison, did you by chance also look into sex-specific differences? Would you expect there to be some?
Yeah, so we also, based on the request, we actually have done some sex-specific genes in there. So you can actually find, on the individual genes that there, we also put whether it's a male or female thing in there. And some of those genes, which we have done both male and female, in finding sex-specific, expression. Which actually we follow the mouse literature, and which we found a very similar differences actually. So we have done some of those. But again, if there's any, yes, I have to do a little bit of advertisement. If there are any gene that you are interested in, but you cannot find on the website, you can actually put request. Then you can just send us email, or there's a message site from that website, so you can ask us to do.
So are there, I haven't been on the website, obviously, are there multiple individuals that are displayed there, or is it, well, you showed this average thing at the beginning, but does the user have access to the raw images of each N?
Yes, they're all, so they're all raw images there. For the numbers, some of them we have done some duplication, but even we say marmoset, we can get a lot of numbers. It's not like mice. So most of them are single. But again, based on the request, we are happy to redo or increase the number. So the database, basically, this is for everyone. It should be useful for everyone's and everyone's research. So we are happy to hear those voices.
Okay, well, thanks a lot for that, and thanks for that resource and for getting the word out. Tomomi, I think we've reached the end of our allotted time. It was really a pleasure. Thanks again for making the effort for joining. Guess you're about to go to bed maybe, but at least it's late in the night, so up to you. And I hope to see you soon in 3D.
Yeah, thank you very much, and also thank you very much everyone for coming. That was great opportunity.
Take care, everyone. Bye bye. See you next week.
Thank you very much. Bye.